pathogenic strains of Escherichia coli. So E. coli is a gram-negative, non-spore-forming rod. It's a normal part of the intestinal tract of both humans and a variety of animals. Most strains obviously are harmless because we carry so much of it in our gut, and it's not like we're walking around sick all the time. But there are some pathogenic strains, and those are highlighted here, specifically these 4 groups. Enterotoxigenic E. coli is pictured here as kind of like cholera in the sense that it just sits on the surface of the tissue and then will produce a cholera-like toxin that goes into the cell and can ultimately cause diarrhea through the same mechanism pretty much. EPEC produces a special binding complex at the surface that rearranges host actin and causes this weird pedestal to form. And so this disruption of the apical surface of the cell can give you diarrhea. And then over here, enteroinvasive E. coli can get in and basically go into a vesicle and survive, and then pretty much move around the cell, like what I've shown you before with Listeria and Salmonella and so forth. And then Enterohemorrhagic E. coli, arguably the worst of all these guys, is essentially like EPEC in the sense that it makes the pedestal and binds the tissue. But this guy also makes a really deadly toxin that can destroy your intestinal lining. So a little bit about enteroinvasive E. coli. They don't produce toxins, but they do invade and multiply inside the cells. And that process of invading and multiplying in the cells can kill the cells and lead to hemorrhaging and dysentery. So again, dysentery is an inflammatory disorder that is the hallmark of which is bleeding in your stool. It can often be misdiagnosed as colon cancer. The infectious dose is quite high, in the millions. So it takes a lot. You have to eat a lot of contaminated food, for example, to get that. E. coli will invade in a manner similar to Shigella. And so here this guy is taken in by an M cell, for example, and then this process of transcytosis where the bacteria are presented to underlying macrophages in the localized immune response. And so these guys should be killed here, but many times they're not. And so if the bacteria can survive, they can actually escape and then invade again through the basal surface of the cell and then do their spreading. And these guys, like Listeria and Shigella, are using this actin tail approach that I've already shown you. EPEC is a bacterium that generally causes infantile diarrhea, so it can spread through hospital nurseries and so forth. But there are food-related outbreaks that have occurred. You can get 2 manifestations. One's a watery diarrhea version, usually associated with the attachment to the epithelium, and then this physical alteration that you see here. So these little microvilli, their job is to absorb nutrients, but when they're all distorted like this, they can't absorb nutrients, and so you get a big buildup of solutes and so forth here, and then water will flow out to meet that higher concentration of solutes. You can also get a bloody diarrhea where attachment and an acute tissue destructive process can take place. It's probably mediated by some kind of toxin, but the research on that is not as well known. Here's a closer look at the EPEC-Cell binding. So these guys are great because they have a few mechanisms at place here. So here's an EPEC-Cell, and I don't know why they drew it like an egg, but they're clearly rod-shaped. But anyway, the microbes have a type 3 secretion apparatus, so this syringe that you see here. And they have a pore protein called ESPD, a tube called ESPA, and again the motor proteins that allow secretion to take place. So EPEC will make this syringe when it comes in contact with the host tissue and then inject a protein called TIR, which goes into the membrane of the host cell. So this is the host cell. This would be your intestinal lining. By putting those TIRs into the membrane, that gives the bacteria an extra receptor to stick to. And so the type 3 secretion system retracts, pulling the microbe into contact. And so the microbe will have these adhesins on its surface, these little green spheres here called intimin. So imagine my pointer dot is intimin. As this guy gets pulled in, to the cell from the retraction of the type 3 secretion system, the intimin binds to the tear and forms this intimin-tear intimate relationship with the cell surface. This process will also, the TIRs, the cytoplasmic portion of the TIRs, will also activate the cytoskeletal elements that you see here, including actin and actinin, and talin, and some of these other things, and cause these guys to polymerize. And so that polymerization pushes up the pedestal. And so again, you can watch that here. EHEC is like cholera. It's also called traveler's diarrhea or Montezuma's revenge. If you've ever gone to some other country and gotten a nice case of watery diarrhea, odds are it's an EHEC strain. It doesn't invade the tissue, it just sits on top and produces a typical enterotoxin. Again, the infectious dose is usually pretty high because they don't survive well in the intestinal tract. You've already seen this figure with cholera. So that's the CT is the cholera toxin. LT is this heat labile toxin, the primary toxin that EHEC produces. I don't know why it says EIEC there. Sorry. And so the EHEC bacterium will produce this toxin. And again, it's an AB toxin, does all the same things that cholera toxin does. I don't know exactly why this toxin, LT, is so much less deadly than cholera toxin, if given that they have the same function. And it may have something to do with how it ADP ribosylates the G protein. There may be a slight difference in the ADP ribosylation. Enterohemorrhagic E. coli, as I said, is the deadliest of the bunch, and it was first identified as a human pathogen in the early 80s, which suggests that it probably evolved relatively recently. It was associated with an outbreak of hamburgers from a fast food restaurant when I was a young microbiologist, and I got really interested in it back then. And so these were undercooked hamburgers at the Jack in the Box, actually, not to disparage Jack in the Box, but that's what happened. So the strain name is typically O157H7. So hopefully you remember from that serotype slide. O for O antigen 157 and H for flagella antigen 7. So there's a variety of flagella and O antigen types of E. coli. It causes a hemorrhagic colitis, which again is an inflammation typified with bleeding. But one of the interesting things that it also does is causes something called hemolytic uremic syndrome, or HUS. And that means that the toxins get into your blood and get filtered out by the kidneys and can start damaging the kidneys. And so individuals often have to go on dialysis. I've seen little kids on dialysis as a result of eating EHEC in undercooked ground beef or in contaminated vegetables. The death rate can be quite high in those individuals. I had a friend who was vegan actually, and developed enterohemorrhagic E. coli from bean sprouts. That's what I think she got it from. Just through an analysis of her diet, I didn't do anything more than that. She was basically sick for about a month and was on dialysis. But 2 to 7% of EHEC infections progress to HUS, and so it can be a pretty serious thing. There was a E. coli outbreak not too long ago associated with Chipotle, and so that killed consumer confidence in Chipotle for a while as well. I don't think it was EHEC though. The origin of EHEC is interesting. This is a microbe that acquires its toxin gene through transduction, just like cholera. And so in transduction, remember that it's a virus that transfers transfers the gene between bacteria. So imagine this virus has infected a bacterium like Shigella that carries Shigella toxin. The virus will infect, chop up the host genome, and then assemble new babies, and each of those new babies gets DNA stuffed into its little capsid, and then the virus goes out into the world to infect something else. But sometimes the viruses make a mistake and will package the DNA from the previous host rather than its own DNA. So some of the viruses will go out into the world with bits of the previous host's DNA, and that's what happened here. The virus infects the new E. coli, injects the Shiga toxin gene, and just like that, you end up with a much more deadly strain. So here's a mild diarrhea-causing strain that acquires this Shiga toxin, and it becomes this super deadly bloody diarrhea kidney failure strain. So again, the hemorrhagic colitis is typified by a bloody red stool. Relatively slow onset. It could be up to a week, and the duration lasts for a while. But what's really important here, because this is all variable, this totally depends on the host and the immune system of the host. But this is really big. Infectious dose is super low for this organism. As you've seen with other E. coli's, it's usually up and over 1 million. But for whatever reason, these guys have the ability to survive the stomach acidity and get into the intestinal tract and cause infections at very low dosages. Just a little bit about the EHEC toxins. We call them Shiga-like toxins because obviously as the bacteria replicate them, as the E. coli bacteria replicate them, they tend to change them slightly. And so they've diverged quite a bit from the actual Shigella toxins that they came from. Not surprising. There's two main types, Stix-1 and Stix-2, and arguably Stix-2 is more toxic. But the important note here is that they act by inhibiting protein synthesis, and I'll show you that here. So this is just showing you a little bit about resistance and sensitivity. Things like animals like cows are probably the reservoir where these deadly bacteria live in the environment without causing symptoms. And cows probably have a natural resistance to them because if the microbes are in there producing sticks, these guys can activate the early endosome, which can then fuse with this, they produce lysosomes, which fuse with the early endosome to form a phagolysosome, and then this gets destroyed. The toxin gets destroyed, and then the microbe will get rid of it. So that's what happens in a resistant animal. In a sensitive animal, you still get endocytosis of the toxin. It still goes into an endosome, but there's typically not lysosomal degradation. The toxin is, for whatever reason, survives and then is shunted through the Golgi and ultimately to the endoplasmic reticulum, and then ultimately can interfere with ribosomes on the endoplasmic reticulum. And so what it's doing specifically is over here. The Shiga toxin can actually destroy the ribosomal RNA that makes up the ribosome of the host cell, of you and I. And by destroying our ribosomes, it prevents us from undergoing protein synthesis, and that actually can kill us and kill our cells. And so just a little bit about it. It's the predominant cause of EHEC-associated disease, obviously, this 0157H7 strain, but there are other versions of this. It definitely produces a lot of Shiga-like toxin, and some of the places we can find it. We know the reservoir is probably cattle, asymptomatic carriers. It can live in the intestines and spread from there. But the foods that we find it are typically unpasteurized milk. I've seen cases where, mothers that think they're doing a great job for their kids, giving them unpasteurized milk because they're convinced that the pasteurization process destroys the nutrient content and makes it less natural. But in turn, again, I've seen small children that have darn near been killed by unpasteurized milk and EHEC infections, among other things. Certainly undercooked beef and other places as well. Like I gave you the example before of EHEC and the bean sprouts. And then, other vehicles include roast beef and apple cider. And when I use the term vehicle in an epidemiological sense, I mean anything like food, water, soil, so it's a medium of some kind, as opposed to a fomite, which is an inanimate object.